we have hit the 12 o'clock hour. Do you want to get us started, Roger?
Yeah, let's do that. All right. So for those of you who have logged on already, welcome to the, I guess it's our first virtual meeting of the Florida section of AWRA. My name is Roger Kopp. I'm president of AWRA this year. And we're excited about having this virtual meeting. We normally have a meeting, about 6 meetings a year, typically every 2 months or so. And we're trying this, and what we may do is we may have meetings on a monthly basis moving forward. And we've got a number of annual sponsors that you saw on the prior slide. These are sponsors that fork out some money at the beginning of the year. Gunster, the Balmoral Group, North Star, and Stanley Consultants, and we certainly appreciate that. It really helps us with our organization, handling our meetings, and then also helping support our students. And then for this particular meeting, we've got a number of sponsors: Hanson, Kimley Horn, Black and Veatch, Clementi Environmental, GHD, and Lewis, Longman, and Walker. And so thank you for our meeting sponsors as well. I wanted to just mention that that we're being recorded, and we're going to post that recording to YouTube, and we're going to put a link to it on our website. So if you don't want to be recorded, I guess maybe turn off your mic and your camera and don't ask any questions. We'll try to make an announcement prior to the next meeting so that you're aware of that. Just so that we have good web service, you know, the data being transferred over the internet, we'll ask that you turn your cameras off until we get to Q&A and mute your sound. But of course, you know, the speakers will appreciate any sort of encouragement, and so, you know, pipe in as you feel appropriate to do. And so we're also going to, so you got to enter your audio pin for the PDH hours for the Q&A, and then for there's going to be a raffle at the end. Yay! In order to do the PDH credits, there's a chat box that's available, and it's in the upper right-hand corner of your screen. And if you click on the chat button, that little thing here, you can send a chat to Amanda and put in your name and your PE, and then we'll be able to record that, and also your audio pin to record your attendance. And so let's see what's next. I think next is I hand it off to Brian.
Okay. And I just want to add to, you know, thanking our sponsors, because by their donations and support of AWRA Florida, we're able to accomplish one of our primary missions, which is to support students and to support them by giving out scholarships. We give out over $20,000 worth of scholarships annually to students going into the water resources field. So the sponsorship is greatly appreciated, especially in this transition time where we're going to the virtual world, making sure that we want to recognize that support. Like Roger said, we want to encourage people to ask questions. So please, during the presentation, if you can briefly post your question to the chat room. Dan's going to monitor that, and then afterwards, he'll go around the room basically as our moderator with the microphone. He'll call your name and hand you the microphone and ask you to directly, please turn on your mic and your video and ask your question of the presenter. So hopefully we can keep our virtual meeting a little more interactive that way. If anybody has any ideas for future meetings on the format, please pass those along to me, and we'll incorporate those as this is new to us, but we think we have most of the bugs figured out. So today we have two fantastic speakers. They're both PhDs in the water science field. Both are studying highly relevant water-related issues specific to Florida, and both are graduates from the University of Florida, so go Gators. First up, we have Dr. Tracy Fanara. She has a unique background in both engineering and in the science realm. She earned her PhD, her MA, and BS in environmental engineering from the University of Florida. Again, go Gators. After 10 years of consulting engineering experience, she joined Mote Marine and currently serves as the program manager for the Environmental Health Research Program, and she is actively developing strategies to alleviate man-made and natural impacts on the environment and public health. So with that, thank you, Tracy. I'm going to ask Dan to turn over control to Tracy so she can go through her presentation.
Hi, everyone. My name is Dr. Tracy Fanara. I'm the program manager of environmental health at Mote Marine Laboratory. And as Brian said, I am a triple Gator and very proud to be. Come on, you can do it. Oh, there we go. So everybody always tells you to start off with any talk with your why. And for me, I can trace it back to two particular moments in my life when that kind of steered me on the path to where I am now. And the first was in 4th grade. I was told about a hazardous waste dump site where industries were dumping toxins into a canal. And those toxins were leaching into the soils and moving with groundwater, and people started building houses in schools. And there were birth defects in cancer clusters. And this is when I realized that everything in the world is connected. What we put into the environment eventually comes back to affect our health. This incident was local to me. It was called Love Canal, and it was the tragedy that initiated the Superfund program. The second time that really kind of threw me forward on the path that I'm on now is when I learned that unsafe drinking water was the leading killer among children worldwide. And that's when I realized that I had found my passion. I wanted to do something whether I was paid for it or not. So to make a really long story short, I became a superhero. I was told about this discipline where I can provide clean water and protect people from natural disasters and design things and make sure that the world had enough food. And I was like, sign me up. So I became one of you. Upon graduating with my PhD, I realized how important it was to have a communication aspect to any kind of scientific discipline. And I had an offer from Mote Marine Laboratory where I would be running a research program that I can do both research and communication. And really that just means that you have 2 jobs. But with that, the program that I was going to be taking over had historically been funded on Florida red tide. Florida red tide, it's a common name for a large population of a phytoplankton, microscopic phytoplankton called Karenia brevis. There were reports of this species since the 1800s. It is native to the Gulf of Mexico. And I went from watershed modeling to focusing on a microscopic phytoplankton. And I was like, oh, all right, how am I going to make this transition? But I started getting the phone calls from the public and how affected people were. See, Florida red tide is a toxic algae species. Over millennia, we've gotten 70% of our oxygen from phytoplankton. 1 to 2% of those are harmful. And Florida red tide is one of those species. What makes this species so unique is that it releases a toxin called brevetoxin. that can aerosolize, meaning that it can go up into the air, attach on sea salt particles with winds, move on shore, and cause respiratory irritation in the individual. But for those with COPD or asthma or any kind of respiratory illness, this can be really serious. And we saw in 2018 especially, but we see this every year. We get a bloom about every year. And we've seen the dead fish and the manatees, the charismatic megafauna, and it just breaks your heart. We've also seen the water discoloration. These blooms are thought to start, the only evidence of initiation of these blooms is offshore at the ocean bottom. And with upwelling, these blooms are brought to the surface, and with currents, they are brought near shore. At that time, when these blooms are close enough to shore, and in large enough numbers to outcompete other species, that's when they may be using surface water nutrients, or they can use surface water nutrients to sustain or exacerbate an existing bloom. Florida red tide and the species Karenia brevis, they're very slow growing. They're outcompeted by other species easily, but what Karenia brevis has that other species don't have is the ability to use over 12 different forms of nutrients. So that's why it's thought that these blooms start offshore, where they have less competition, but they can utilize other forms of nutrients that other phytoplankton can't. So when I got to Mote, I was like, okay, obviously the problem is that they haven't had an engineer on this. I'm just going to go in there and solve the problem and stormwater. But I was just so humbled by the complexity of our marine ecosystems. And so at Mote Marine Laboratory, to deal with this complexity and this difficult topic to research. I mean, we're talking about a microscopic phytoplankton body of water that acts differently in a lab than it does in the natural environment. We have 5 different programs at Mote Marine Laboratory working on Florida red tide research. We have AUVs, we have in situ remote sensing. This AUV that you see here, it is commonly called a glider. It goes into the water for long periods of time. Every time it has a number of sensors in its payload. Every time it surfaces, that data is transmitted to our computers, so we can hopefully get a rapid response when we get a chlorophyll signal on the bottom of the ocean or anywhere in the water column that we didn't know was there. And as I was saying earlier about bloom initiation, as of right now, the only evidence that we have is that these blooms start in the ocean bottom, and with upwelling, they're brought to the surface. But the interesting part is how it gets that nutrients being offshore. So I don't know if you've seen Weather Channel recently, but there has been a lot of talk about the Saharan dust. The Saharan dust coming over from Africa into Florida. Apparently today there was a heavy haze from Saharan dust in Tampa. Yeah, so this Saharan dust.
Everybody, please mute your microphone. We're getting a lot of background noise coming through into the webinar. Thank you. Go ahead, Tracy.
Okay. So the Saharan dust coming over into Florida is bringing with it, first of all, phosphorus, which it drops into the rainforest. And it's the reason why the rainforests are so diverse and amazing. But it also brings over iron. So it's thought that these blooms can potentially initiate because the Saharan dust is bringing iron over to a species of cyanobacteria, which is a nitrogen fixer. This species is called Trichodesmium. It's a marine cyanobacteria that can take nitrogen from the atmosphere. So it doesn't need nitrogen runoff from surface water. It can get its nitrogen right there. So it is more likely to be offshore in those low nutrient waters where less competition is there. So when these Saharan dust bring the iron over to the Gulf of Mexico, the trichodesmium can then bloom using that iron, that limiting nutrient, to proliferate. And then upon decomposition of that species, it's thought that that's where Karenia could be getting some of its nutrients and then multiplying from there. So we have a bloom every year somewhere in the Gulf of Mexico. These are just some blooms that we've had throughout the years. And of course, this is the one that well, the beginning of the one that everybody remembers. In 2018, everybody in Florida remembers this. Florida was in a state of emergency due to 2 harmful algae blooms. Both the Florida red tide bloom that started in November 2017 and then intensified in August 2018 following heavy rainfall, among a number of other things. And it's thought that a second bloom actually met up with that first bloom in July, August 2018. And then we also had a cyanobacteria bloom in Lake Okeechobee, a big lake in the middle of the state. Due to heavy rainfall, Lake Okeechobee needed to release this water that was laden with cyanobacteria into the Caloosahatchee River. That water then outfalled into the Gulf of Mexico, where we had an existing Florida red tide bloom. Now we had increased intensity of Florida red tide simultaneously from Manatee County all the way down to Collier County. So what this means is, of course, you know, Florida red tide can use over 12 different forms of nutrients, and number 13 is actually Microcystis, the harmful species that was predominantly in this cyanobacteria bloom. So Karenia brevis could have very well used that decomposing cyanobacteria as a nutrient source. However, we already know that there was lots of nutrients coming in from Caloosahatchee River, but we also know that there was a lot of nutrients coming in everywhere throughout the coast, from septic, wastewater overflows, from stormwater, because we did have intensity increase throughout the coastline. Oh, and just to clarify, I should probably do this. Cyanobacteria is a freshwater species. Cyanobacteria, there are thousands of cyanobacterias, but the one predominantly was a freshwater species called Microcystis. And it doesn't do well in saline waters. And Florida red tide, Karenia brevis, is a marine species. And a lot of people ask the question of these storm events and all of this stormwater coming in, what it does to red tide. And yes, it can provide that nutrients, but it also decreases the salinity. And one of the things that I'm researching is at what point does that increase nutrients and that decreased salinity lyse or exacerbate an existing bloom. What we learned from that point was that communication was never more important. In fact, it was the most important aspect at that time. We were, there was a lot of misinformation, with 90% of Americans getting their news from social media. It was really difficult to combat because the loudest voices were not scientists. So we had a huge obstacle to overcome. So we had to think fast, and especially in my program, because I'm focused on a lot of that communication about Florida red tide with the public. So these are some things that I did. First of all, getting communities involved, giving people ownership, empowerment, that they are part of the solution and they're with scientists. We're all on the same team. Teach people just how the world works so that they are better ready to understand the complexity of Florida red tide, and tell people what they can actually do themselves, how they can make changes to improve our water quality. So these are some of the projects that we have. We implemented a number of different citizen science projects. these are some of the ones that we already have citizen scientists working on, and I've listed some of the ones that we have just gotten funded through the Red Tide Initiative focused on mitigation of Florida red tide blooms. The BCRS is one of the things that was most popular. We had over 1.6 million users on our smartphone app and website. This was first developed in 2006. I redeveloped it in 2015. We have 40 beaches reporting anything that anyone would want to know when they're going to the beach: crowds, surf, debris, jellyfish, but also Florida red tide effects such as respiratory irritation, dead fish, and water discoloration. These are an example of some of the reports. Our reporters are trained beach sentinels, lifeguards, maintenance, and they are able to upload pictures daily. We also have links for information about red tide, especially for tourists when they come down, to really fill in the blanks between those beaches, because Florida red tide is patchy. So you might experience respiratory irritation at one end of the beach and not at the other. So we want as many reports as possible so that people get that information in real time and can make decisions on which beach to visit on a regular basis, so that they can have a healthy and happy experience. When someone comes from up north, has a bad experience at the beach, they think that that's how it is all the time. So CSIC, just wanted to make the acronym work, get it, CSIC. It's a citizen science reporting app where anyone can report from anywhere in the world environmental hazards, including red tide effects of respiratory irritation, water discoloration, and dead fish, if you're geolocated in Florida or up the East Coast. HABSCOPE is a NASA-funded project that we worked on with NOAA and GCUS. We trained our volunteers to take samples of water, put it underneath a cell phone microscope, upload a 30-second video of their sample into an app that has an algorithm that automatically calculates the concentration of red tide by shape, size, and movement. That information then goes to a NOAA respiratory irritation model. We're also working with artificial reefs, oyster reefs, trying to see what kind of clearance rates these filter feeders may have. Now, we've seen that a lot of these artificial reefs are mainly colonized with tunicates and barnacles, sea squirts, and the ones in the. If you want to know more about tunicates and barnacles, please email me. I have lots of information. But these aren't the most effective filter feeders. Mussels and oysters are, but only sometimes do they colonize on these structures. So we're trying to see how well these can do in the canalways where people don't have a choice to leave their home and are being affected by the respiratory irritation of Florida red tide. We're trying to see if biofiltration can be a promising way to alleviate those effects. Florida red tide would be here whether we were here or not. These blooms would be happening. But the question is, what role do we play in the exacerbation and prolonging of these blooms? And so I always add this into my talks just because this is what I did my dissertation on, hydrologic restoration. I was in stormwater design and treatment for years. So I always kind of add this at the end. And you guys, being engineers, you know how the water cycle works in development and engineering. So I'm going to go through this really briefly. Basically, we have manipulated and changed the water cycle by putting impervious pavement down on our land, implementing stormwater pipes, having the water run off fast at lower water quality, causing erosion and algae blooms, among other things. So my dissertation was actually to go back into urban environments and see what we can do to retrofit these environments to mimic the water cycle, treat nutrients at the source, and eliminate or limit the occurrence of cyanobacteria blooms freshwater. So this is just an example of one of the projects, and actually Natalie, that is speaking next, worked on this project with us. And this is kind of, I usually do these talks for the public, and people are always asking what they can do right now. And I always tell them that when the rain falls on your roof, it goes into a gutter, down your driveway, into a storm sewer, right into a natural water body. What you can do right now is disconnect that impervious surface by implementing a cistern and a rain garden or infiltration trench, xeriscape, things like that, to limit the amount of fertilizer that you're using and increase the amount of native plant species that you have in your yard. And this is information on where to find information on Florida Red Tide, and also where you can get a hold of me if you have any questions, or if you're on social media.
All right, thank you, Tracy. She also has a whole lot of other interests, and you can see Inspector Planet there. She runs that. I believe that is type of a YouTube channel where you make a lot of videos with educational material related towards science. Also was on the television show Mythbusters for a period, and I've seen, if you do a search for Tracy, you'll see she's been on television quite a bit, does a lot of interviews. So there's a lot of material there. I have definitely abbreviated the bio down quite a bit.
Weird Earth is on Weather Channel every Sunday night, and What on Earth, right now they're playing reruns on Science Channel.
All right, Dan, do we have any questions that we can ask the audience to bring up for Tracy?
Just having a look here.
Okay, I'll throw out a first question that I had during your presentation, Tracy. I know you talked about the Saharan dust cloud that typically comes over across the Atlantic. Today we are supposed to be receiving the start of a really large one that's been coming across for the last at least several days, if not weeks. Do you expect this to have a significant impact this year on red tide?
That's a really good question. I mean, research has shown that it's had an impact on Florida red tide in the past. Well, this year, I mean, we do have an upwelling event that is occurring as well. So our hypothesis is that likely, but we never can say anything for sure. There are so many moving parts and so much research to yet still do. I mean, think about hurricane tracking. We still don't know exactly where it's going to go, and think about the amount of energy, money, and time that has gone into that compared to Florida red tide. A small fraction, probably less than 1%, it has gone into that. So it's understandable that we aren't able to predict these blooms yet, but we're hoping that with advances in research, we will be soon.
Okay. Do we have any other ones, Dan?
Yes, we have one more, just asking if we know what causes the upwelling in terms of like the timing of that upwelling. Is it some kind of circulation change, or?
Yeah, upwelling events happen when denser water, I mean, when the density of water change, when water gets stratified and then those waters, more dense or less dense waters come up to the top. So that happens anytime that we have changing of the seasons. We get stratified, the top water's warm, and it kind of makes layers within the water column. And that's why you see a lot of algae blooms during this time too, because a lot of species have a cyst phase, and Karenia brevis is hypothesized to have a cyst phase, even though we haven't seen it. And when these cells then get exposed to this stratified layering in the water column, that's when they experience a change in environment and a trigger where the cyst is actually opened. We saw this in California with that bioluminescent bloom that made national news. That is a known cyst phase. So upwelling occurs generally when water temperatures change. Or, you know, we had that forced upwelling event with that, basically with that hurricane. So a lot of NASA has correlated really intense Florida red tide blooms with hurricanes preceding those events. like Irma, we saw that water just come off the coast of Tampa, and it's hypothesized that that may have been a forced upwelling event that then sparked that really long, intense bloom.
Thanks, Tracy. We have another question here. Taylor, do you want to unmute yourself and ask that question? Taylor Willis has a question. Okay.
I can read it.
Oh.
No,
There
no,
you
go
are.
for it.
Okay. What are your thoughts? What are your thoughts about the phosphatic clay being used in places such as China for red tide?
So the phosphatic clay, we did it was before my time. Mote Marine Laboratory did a study with Woods Hole back in like 2004 with the phosphatic clay, but the amount of clay that was needed was, it worked, but it posed threat to benthic ecosystems because of the amount that they needed. Now, there is a new clay that was actually made in China that they're using for red tide that is not phosphatic clay. It's actually absorptive clay coated with aluminum chloride, aluminum oxide coated, that is a better flocculant, just like in a wastewater treatment plant. It can actually flock the Cornea brevis cells and cause that to settle, the flock to settle. That's one of the things that we're testing right now in the laboratory and doing mesocosm studies on that with Woods Hole again. And it's seeming that it's very promising. Now, it again, like apparently, well, not apparently, we know that there's a lot less that we need to kind of control a bloom volume-wise with this product. Whether our studies will say that it's, doesn't pose threat for benthic ecosystems, we haven't got there yet. But we do know that this is more promising product than what was previously tried with the phosphatic clay. Does that answer your question?
All right. Well, thank you, Tracy. For time, we need to wrap up here, but thank you very much for the presentation. It's great information and definitely relevant to today. Our next speaker is Dr. Natalie Nelson. She is an assistant professor in the Biological and Agricultural Engineering Department at North Carolina State University. She received her BS and PhD from the University of Florida. Go Gators. During her graduate studies, Natalie was active in AWRA. She received the Richard A. Herbert Memorial Scholarship, and that was a scholarship from the AWR National Organization. And she also received several scholarships from AWRA Florida, our own organization here. In 2016, she received an award for graduate student for the statewide research poster contest. In 2014, she received the Sanford N. Young Scholarship, and in 2013, the William V. Storch Student Award. So she was also president of the UF Student Chapter for AWRA, and under her leadership, they won the Outstanding Student Chapter Award that year. So Dr. Nelson is a prime example of the type of students that we try to support, and we love the fact that now she's here as a professional and giving a presentation. So Dr. Nelson, go ahead.
All right, thanks so much, Brian, for the introduction. And I really owe Florida AWRA a lot of thanks for their support while I was in graduate school. So it's really special to me today to have the opportunity to be able to connect, this time from the comforts of my living room in Raleigh, but it's nice to be able to engage with you all. And I should also say thanks to Brian and Dan for coordinating the webinar today. And also, I think I owe Jeremy a thank you, because I'm guessing he's probably who threw my name in. All right, now let's make sure I have control of the screen. All right, perfect. So today I'm going to be speaking with you all about a project we have funded through the National Science Foundation, through which we're looking at connections between water chemistry, phytoplankton communities, and red tide in the Caloosahatchee Estuary. And I want to acknowledge my collaborators, Dr. Edward Fliss with the University of Florida and Dr. Eric Milbrandt with the Sanibel Captiva Conservation Foundation. So we're focusing on the Caloosahatchee Estuary, located on the southwest Florida shelf. And we're particularly interested in the Caloosahatchee Estuary because it's located in an area where we see a collision of a lot of different factors related to harmful algal blooms. So the Caloosahatchee Estuary has its own watershed, but it also receives waters from Lake Okeechobee. So proportionally, in terms of the discharges out of Lake Okeechobee, we see the largest fraction going to the Caloosahatchee Estuary. And Lake Okeechobee is a highly eutrophic lake. So with the discharges that come out of Lake Okeechobee, we also see a lot of nutrients in terms of the dissolved inorganic nutrients, but also organic nutrients coming through those flows. Lake Okeechobee also regularly experiences blooms of cyanobacteria. So also with those discharges from the lake, we can see export of cyanobacteria blooms and then those blooms being transported to downstream waters. The Caloosahatchee Estuary is also located in this area that we think of as the epicenter of red tide impacts in Florida. So in particular, the bloom that we're all familiar with, the red tide that lasted from 2017 to 2018, was particularly severe in this area around the Caloosahatchee Estuary. And like Tracy described in her presentation, there are certain processes that we do understand fairly well about Karenia brevis. So we know that they initiate offshore and they're transported to the nearshore environment. But once they're in the near shore, we see that these red tides can last for a really variable amount of time. Sometimes it's pretty short, sometimes it's longer. And sometimes those durations can be explained by different coastal circulation processes, but other times it's not really clear what exactly causes that longevity. So one question that's been popping up more recently, and it's been around for a while now actually, is what is the role of fresh water runoff and transporting nutrients that could then serve as potentially nutrient fuel for red tide in the nearshore environment? And these kinds of concerns have been really nicely summarized in a recent report that was published by Florida Sea Grant. So in August 2019, there was a symposium, the Florida Harmful Algal Bloom State of the Science Symposium, where a lot of experts were convened, very likely multiple people on this call, to identify different issues that were basically like knowledge gaps in terms of what we do and don't understand about different harmful algal blooms that occur in Florida. And here I've just taken a screenshot from one of the pages in this report where they summarize some questions that we don't really have the answers to. And these questions are related to the initiation, maintenance, and termination of red tides. So I've put here in boxes 2 different sets of questions that I'm not going to read out directly, but I'll summarize them as basically being that we have a lot of outstanding questions with regards to the interactions between land source freshwater runoff and other kinds of phytoplankton, particularly cyanobacteria, and what their connections are with Karenia brevis. So with our, there's one thing I should mention on this last slide actually. One point in particular that's raised is that part of the reason why we're really interested about connections between cyanobacteria and Karenia brevis is that Karenia brevis is known to be mixotrophic. So one of the questions that's listed here is, what is the role of mixotrophy? So for those of you who might not be familiar, mixotrophy is a characteristic of organisms where they can be phototrophic and heterotrophic. So there are certain kinds of algae that we know have this mixotrophic characteristic, and Karenia brevis is one of them. So what this means is that Karenia brevis can get their energy from photosynthesis, but they can also get energy by ingesting other phytoplankton. So they can take up different kinds of nutrient sources and they can graze on other phytoplankton if they need to supplement their energy if it's not being met fully by photosynthesis. So this is also what Tracy was describing in her presentation. But because they can do this, that implies that then if you had, say, cyanobacteria that were discharging to the coast, that the Karenia brevis could then potentially use those kinds of phytoplankton as an energy source. So when we're thinking about nutrient fuel for Karenia brevis, we can't only think about dissolved inorganic nutrients, we also have to think about the other kinds of phytoplankton that are present. And so although we know that P. brevis is mixotrophic, what we've seen is that in terms of field analyses that really try to capture the potential for Karenia brevis to be grazing on other kinds of phytoplankton, we don't see as many studies in that area. So what we were interested in doing as part of this project is first checking to see, well, how do nutrients vary from the head of the estuary to the offshore environment in relation to red tides? And then we also wanted to determine if phytoplankton biomass could serve as a key driver of nutrients and carbon or a key source of nutrients and carbon to Karenia brevis. So to get towards answering these questions, we went and collected data in the Caloosahatchee estuary. So we had data across, we collected samples at 13 different sites, going from the head of the estuary all the way into the marine environment. And so those sites are shown here in the black circles. And we can categorize these different sites as being in the upper estuary, the lower estuary, and then the marine environment. So at each of these sites, Eric Milbrandt and his crew at Sanibel Captiva Conservation Foundation, they take out 2 boats to try and get the samples across these different areas at approximately the same time. And we collect samples. Some of those samples would be sent to a local lab for nutrient chemistry analyses. And then the other samples would be sent up to Gainesville at Ed Flips' lab, where he would characterize the different kinds of phytoplankton species that were present across all 13 sites. And we only had enough funds to collect data at 10 different sampling points. We would have loved to collect more data, but we were restricted. So we ultimately were able to collect data across the 13 sites on 10 different dates. So those dates are shown here, but they're also shown in the context of the hydrologic conditions that were present when we collected these data. So on the top here, we have a hydrograph showing flow over time from 2017 through 2020. And the points that you see are the flow observations as observed at S79. So S79 is the lock and dam that's located at the head of the Caloosahatchee estuary. It's where the C43 canal, shown here on the bottom, connects up with the Caloosahatchee estuary. So with S79 flows, you can get a sense of how much flow is coming in from the C43 basin and also Lake Okeechobee discharge. Then in this shaded area, we're also showing the amount of discharge coming out of Lake Okeechobee. So these flows were measured at S77, which is the lock and dam that's essentially at the head of the C43 canal. And then the points are colored yellow if the majority of the flow that was entering the Caloosahatchee estuary was coming from the C43 basin, and then the points are blue if the majority of the flow was coming by way of Lake Okeechobee discharges. So we can see that in particular in 2017, we had these really large peaks that had occurred in flow due to really large Lake Okeechobee releases. So we were able to collect data at each of these time points shown as vertical lines, these vertical dashed lines, and then the color of the lines just corresponds to what the source of fresh water was coming into the head of the estuary. And we also have red arrows shown at the top to indicate when there was a Karenia brevis bloom present in the marine environment. So what I really want to communicate here is that we collected data across a range of flow conditions. We would have ideally had data collected in this large peak here, but we were able to collect some data in this one high flow period, and we also collected data across times when there was a red tide event and there wasn't a red tide event. We were able to cover a pretty nice range. So now looking at some data. So here we're looking at plots where we have sample date on the x-axis. And so here, remember, we collected data from 10 different time points, and they're shown here as being equally spaced, but they weren't actually equally spaced in time. And then we have chlorophyll on the top, and then we have different nutrient species on the other panels. So here I'm showing averages and then the ranges of values that were observed across these 3 regions of our study area. So when you see the green dot, that's corresponding to the average value for the upper estuary. The teal triangle corresponds to the lower estuary, and then the blue squares correspond to values observed in the marine environment. So now first looking at chlorophyll A shown here on the top. So the main things that I want to point out are that in our first 2 time points, we observed relatively higher chlorophyll A in the marine environment relative to the upper estuary. So shown here as those blue squares above the green circles. And then here, this red box is showing when the Karenia brevis events were occurring. So we see that chlorophyll A was relatively higher during these time points, but then for the rest of the time points, for the most part, chlorophyll A was fairly consistent across the upper estuary through the marine environment. There were still some differences, but they were relatively comparable. Next, if we look at nitrogen, so here we're showing ammonium, NOx, or nitrate and nitrite combined, and then total kildol nitrogen. So here the trends were more interesting because they weren't exactly as we expected. So during the Karenia brevis bloom, we can see that ammonium was quite low in the marine environment and then also in the upper estuary. But then after the die-off of the red tide event in 2018, we started to see this uptick in ammonium that was occurring in the marine sites. So what we can see here is that in the marine sites, consistently, ammonium was much larger or much higher in magnitude relative to the upper estuary and lower estuary. In contrast, for nitrate and total killed nitrogen, it was the opposite. So we saw higher nitrate and TKN coming in through the upper estuary, decreasing as we went down estuary, and then being relatively low in the marine environment. So this implied that there was probably phytoplankton uptake of these species of nitrogen coming. As you go down estuary, you have greater uptake of, in particular, nitrate. Then for phosphorus, here on the bottom, we saw a very similar trend to what we saw for nitrate and TKN. So for orthophosphate, we saw higher orthophosphate coming in through the head of the estuary and then decreasing as you went down estuary. Again, that indicates that there was phytoplankton uptake occurring as you go down the estuary. So this indicates that sometimes when we're measuring nutrient concentrations at the head of the estuary, we can't assume that the same concentrations are coming out the outlet because there's quite a bit of uptake by phytoplankton occurring. We can also look at these data in a slightly different way, just to really emphasize some of the spatial trends we're seeing here. So if we look at the trends in chlorophyll A and also nutrients across the transect, so here I'm showing observations from the head of the estuary, going down the estuary and over into the marine environment. So just for these sites. So here then the sites are shown on the x-axis, and we're always going from the head of the estuary to the offshore as we move right. So each of these lines, the gray line corresponds to different sample points, but then the blue line shows us the overall trend. And what we can see here is that chlorophyll A, again, was just generally speaking across our sample points, we had kind of consistent chlorophyll A going from the head of the estuary to the marine environment with some clear variability. But then for orthophosphate, total phosphorus, TKN, and nitrate, we saw a decline, this really strong decline that occurred in these different nutrient constituents as we went down estuary. But then the opposite trend was observed for ammonium. So you see this uptick that occurs as you go into the marine environment. And there were a few cases when ammonium was 0 in the marine area, and that was because of the presence of the Karenia brevis bloom. We can also look at these nutrient trends in terms of what they might mean for phytoplankton and nutrient limitation. So if we look at the molar ratio of total nitrogen to total phosphorus, we can see what the trends are in terms of whether or not the phytoplankton would have sufficient nitrogen or sufficient phosphorus. So basically trying to identify which of these nutrients might be limiting their growth. So when we see the total nitrogen to total phosphorus ratio greater than 20, that indicates that the phytoplankton are phosphorus limited. And then when we see it lower than 20, that indicates that the phytoplankton are nitrogen limited. And interestingly, during our sample points, during our field campaign, we consistently saw phosphorus limitation. So here we're showing those values in the upper estuary, and this dashed line is corresponding to that 20 threshold. Here you see the values for the lower estuary, and then here for the marine environment. So typically what's been reported before is that in the upper estuary, you have strong phosphorus limitation, and then as you move down towards the marine environment, you typically trend towards nitrogen limitation. Well, what we show here is that actually we saw much more severe phosphorus limitation during our 10 observation time points. So this is in contrast to what's previously been reported. So I mentioned at the beginning that we had these two motivating questions. One is, how do nutrients vary from the head of the estuary to the offshore environment in relation to red tide? Well, what we saw is that nitrate and orthophosphate were all very low in areas affected by these Karenia brevis blooms, and that ammonium was also low during the 2018 red tide. So this implies that there was really strong nutrient limitation occurring in that marine area around Sanibel Island during the red tide event. And when we see really strong nutrient limitation, that indicates that we're more likely to have instances when those Karenia brevis are going to start to ingest other kinds of phytoplankton. So that to the point where the nutrient supply is low, they need to start moving towards alternative sources of nutrients, such as then grazing on other phytoplankton. So the conditions were such that we might expect to see some of this mixotrophic characteristic actually shown in the Karenia brevis. And then outside of the 2018 event, we saw that ammonium was relatively high, and we're still trying to understand why this might be. So we know that there was a lot of mortality that had occurred because of the red tide in 2018. So we would have expected to see kind of an uptick in ammonium that would occur due to the remineralization of the biomass that would have died off because of all the wildlife that had died due to the red tide. But we wouldn't expect that decomposition to persist for so long such that we see ammonium be high up to a year later. So we're still trying to understand what might be happening here. So we think that because we saw this really strong phosphorus limitation in the marine environment, it's likely due to the fact that there was this surplus of ammonium. So if any of you have ideas as to what the ammonium source might be in this case, I'd be really eager to hear. All right, so now moving on to some of the phytoplankton. So I mentioned that we think that because there was really strong nutrient limitation, that it's possible that also these Karenia brevis are munching on other kinds of phytoplankton. So here what I'm showing is pie charts that show you how the phytoplankton community was composed of different types of phytoplankton across our 10 different sample points. So each pie chart shows you the relative contribution of these different classes. I'm not going to go into much detail here. My main message with this particular animation is just to show that the community changed a lot throughout those 10 different sample points. And that is what we'd expect. It's a dynamic system. We know that the phytoplankton community responds strongly to these different changes in environmental conditions. But what we did notice is that here, so Picocyanobacteria is shown in teal, and then Karenia brevis is shown in red. So we see that in the instances when we had Karenia brevis, the pie charts get bigger if there was a lot of phytoplankton present. You see that when the Karenia brevis was present, there was usually a lot of it. And then we also see that Picocyanobacteria, that teal color, is pretty much consistent throughout all of our sample points across all sites. So it was really ubiquitous. So if we focus in then a little bit more closely on what was happening across those 10 sample points across all of our sites. Here we can see we have we're looking at the amount of Picocyanobacteria shown in teal and then Karenia brevis shown in red across those 10 different sample points and across the 13 sites. So we have only a limited number of observations here, so it's important to keep that in mind as we interpret these results. But for one, what we can see is that Picocyanobacteria were consistently present, and then we only had Karenia brevis present at the first two and last two sample points. And the reason why I'm focusing in on Picocyanobacteria here is because Karenia brevis actually isn't a very large species. So their cells are not that large. And because their cells are not that large, we expect that when they do graze on other phytoplankton, they're going to graze on the really tiny phytoplankton. So, you know, a mouse wouldn't try to eat a live elephant. We're trying to make sure that we're considering what's biophysically possible. So Picocyanobacteria are very small-celled phytoplankton, and they seem like the best candidate in terms of what we might expect for Karenia to graze on. So here we can see some of these trends in terms of how Picocyanobacteria is always present, but in different magnitudes, and then we see how Karenia brevis also changes with time. So we were interested, though, based on the data we have, do we see that when there's an increase in Karenia, do we tend to see a decrease in Picocyano? Because that might indicate that there is this kind of grazing relationship happening. So here what I'm showing is on the y-axis, we have our correlation coefficient, and then on the x, we have the different nutrient species that we considered, and then also different phytoplankton groups. And we're particularly interested in negative correlation. So we want to see what factors are decreasing as Karenia increases, because that would indicate that Karenia is consuming maybe certain nutrients, certain phytoplankton, and then as they're consuming those energy sources, they're starting to grow. So we want to see a relationship where we see this inverse relationship or a negative correlation. And that's what we're showing here with nitrate, phosphate, and then also to a similar extent, Picocyanobacteria. So again, there's only so much you can infer from correlations, but we do think that it's compelling to see that with Picocyanobacteria, these data collected out in the wild, messy conditions, nothing was controlled, you know, it's field data. We can see that there was this comparable negative correlation to what we see for the inorganic nutrients that we would have expected to play a role in fueling Karenia brevis. So from these results, we do think that there's an indication, or at least there's some promise that it would be worth investigating this area further. So then for that second motivating question, could phytoplankton biomass serve as a key source of nutrients and carbon to Crenea brevis? We think that what we've shown with the correlations indicates that it's definitely an area worth pursuing further. It takes a lot of resources to monitor picocyanobacteria because in our case, we're using an expert who creates slides, looks under a microscope, has to actually use their expertise to identify these different kinds of phytoplankton, but there are now new kinds of technologies that allow for this type of monitoring to be done a bit more automatically. So we think that this could be a compelling area to explore further, particularly for those who are interested in really getting to the answer of this question of what's fueling Crenea brevis in the nearshore environment. So our next steps with this project are to start to perform some simple process-based modeling to further explore these relationships and maybe have some stronger conclusions. And then we also want to complement our project data with data that have been collected through long-term monitoring programs through the district. That's all I have for today. Thanks again for the invite, and if there are any questions, I'm happy to answer them.
Okay, thank you. Thank you, Dr. Nelson. I think I'm looking in the chat box. It looks like we do have one question from Don. Professor Duke, can you unmute your microphone and ask your question? Maybe that's not what.
Took a minute. Am I on now?
Yes.
Oh yeah, we hear you.
Okay, thanks. Hi, Natalie. Great to hear from you. And what a great talk. Now I have to go back and read my question to see what it is that I asked. Really interesting that you found it to be phosphorus limited, and especially because so many of our BMPs and sort of response plans have been focused on nitrogen. How do you think that the phosphorus limiting is a routine condition that you captured here? Do you think it was a happenstance that you happened to catch it at the wrong time and that it's usually nitrogen limited? And how many times should we repeat your study to find out the answer to that question?
Yeah, that's an excellent question. I mean, a lot of the prior research, I think, makes a really compelling case as to why we would expect and why we've observed nitrogen limitation in these areas. So I definitely don't want to discredit that work. And I do think that that's typically the case. So what I'm not sure of, though, and what I don't think, I haven't personally seen a study that really looks at when these red tides are ongoing, what do we see in terms of the local water chemistry? Because I suspect that the sort of traditional paradigm of upstream, you have phosphorus limitation, and then in the marine environment, you have nitrogen limitation. That's probably upheld most of the time, but then it seems like when we had this really severe red tide in 2018, the conditions were just so limiting in terms of the nutrient supply that I think that we did start to see this phosphorus limitation occurring, and whether it was connected to what we saw with remineralization of dead biomass, I'm not sure exactly. And I think that probably the more we can understand about why we saw an increase in the ammonium, the more likely we are to understand if this was an anomaly or if this is something that is more of a typical dynamic when you have red tide.
All right. Well, in order to maintain our time and everybody's commitment, I'm going to have to wrap it up there. Thank you very much to both of our speakers today. I think that's definitely some very relevant and interesting information that is useful to our group. I'm going to turn it back over to Roger. I think we have a raffle winner that he can announce.
Yeah, let's see. Did someone send me the information on the raffle? How are we going to do that?
You should have it in your chat, Roger. It came from Amanda.
Okay. Here we go. Okay, it's number 45, Mark Walsh.
All right. And what does Mark win?
Mark wins.
I see it on here. He gets an Engineering with Nature Atlas and a AWRA, one of those custom Columbia shirts that we have. Very good.
Yeah, those are awesome shirts. And what you can do is go to the website and you can, pick out the color you want, the size you want, and I think there's a couple of different styles of shirts, short sleeve, long sleeve, and we will provide you more information on that because you're not going to have to pay for it. So we'll get you some information on how to do that, so we'll make that happen.
All right, perfect. Do you have any other closing comments, Roger, before we let everybody go?
Well, I wanted to say thank you for everyone who has come on today, and we really would like to get any thoughts that you guys have on how this went, any suggestions for how we can do this better, and then just check out our website. We're going to be having another meeting in August, and we're trying to set that up right now, and we're going to have some very interesting presenters, and we're going to have some prizes at that one too. So please check, and we'll also email everybody that we can that joined this one to let you know when we've posted information.
All right, great. With that, we'll wrap it up. Thank you, everybody.
All right, thank you.